To start off, let's look at the cell's size and structure. Both plant and animal cells are what we call eukaryotic cells. All eukaryotic cells have a membrane-bound nucleus and lots of membrane-bound organelles, which are smaller components of the cell that have specific jobs like keeping the cell alive and functioning. Let's talk about some organelles that both plant and animal cells share by starting from the center and working our way out. We already mentioned that both cell types have a nucleus that stores the cell's genetic information. The nucleus is surrounded by a porous bilayer membrane called the nuclear envelope that selectively allows proteins and DNA in and out of the nucleus. The nucleus is surrounded by rough and smooth endoplasmic reticulum in both types of cells. The rough ER is rough only because it harbors another type of organelle called ribosomes that act as sites for protein synthesis. The cell can use these proteins for carrying out genetic processes, sending signals, and for providing structural support for the cell. Smooth ER contains enzymes that processes lipids or fats for cell use. Both cell types also contain the Golgi apparatus, which is responsible for modifying and sorting all kinds of proteins from the rough ER. We can think of this like a shipping and receiving center for mailing packages. Both cell types also have peroxisomes in the cytoplasm that break down fatty acid chains and lysosomes that help break down other molecules so that they can be recycled and reused throughout the cell. The last common organelle between plants and animals is mitochondria. Mitochondria are responsible for energy metabolism by generating ATP. We'll talk about that a little bit later. All right, we've made it to the outskirts of the cell. Although the cell membrane isn't really an organelle, it is the last similar physical feature of plant and animal cells. Just like the nucleus, both cell types are surrounded by a membrane that's known as a phospholipid bilayer. Now that we've gone over some physical similarities, let's talk about some differences. Most notably, the cell membrane of a plant cell is surrounded by a cell wall that makes it rigid and helps the plant keep its form. The cell wall is also important because it has cell junction sites called plasmodesmata that connect one plant cell to another. Animal cells have a similar feature called desmosomes. In terms of organelles, plant cells have a few unique components. Chloroplasts are probably the first thing we think of when we think about what makes plant cells unique. Chloroplasts are the site of photosynthesis, where the sunlight, carbon dioxide, and water are turned into energy for the cell to use. Another organelle is the vacuole. Vacuoles usually take up a large part of the available space in the cell because they store water and other nutrients for the cell to use. If water is scarce, the vacuole will contract and the overall cell size will shrink as well. Animal cell size varies a lot depending on the tissue type, but overall, animal cells are smaller than plant cells. Now that we've gone over the similarities and differences of both cell types in terms of their structure, components, and size, let's talk about how each cell type metabolizes energy. Both cell types directly or indirectly need light for producing energy. Plant cells and animal cells both need energy to carry out cellular processes, but they metabolize energy in different ways. Like I mentioned before, plant cells use a process called photosynthesis, which occurs in chloroplasts to convert carbon dioxide, water, and sunlight directly from the atmosphere into usable energy in the form of glucose. Let's look at the equation for photosynthesis. This is a balanced equation for photosynthesis. When we take 6 carbon dioxide molecules and hydrolyze them with 6 water molecules and photons from the sun, we get a usable form of energy in the form of the sugar glucose, along with 6 molecules of oxygen that get released into the atmosphere. So when an animal eats a plant, the animal is getting that glucose from the photosynthesis reaction that it can use in its own energy metabolism. This way, animals are indirect consumers of light energy. Animal cells essentially undergo the reverse process. Animal cells require glucose in order to gain energy in the form of ATP. There are a few processes that animals can use to generate ATP, but the main process is through an oxygen-dependent reaction called cellular respiration. Cellular respiration is made up of 4 main processes: glycolysis, the Krebs cycle or citric acid cycle, electron transport through the electron transport chain, and ATP synthesis. So, When an animal eats, the glucose molecules in food will undergo glycolysis in the cytoplasm to convert one glucose to two molecules of pyruvic acid to be used in the Krebs cycle in mitochondria. In a bridge step, the pyruvic acid enters the mitochondrial matrix and binds to coenzyme A to form acetyl coenzyme A. It's then transported to the mitochondrion. Acetyl-CoA, acting as a fuel source for the Krebs cycle, undergoes a series of oxidation steps. Between the two processes of glycolysis and the Krebs cycle, 6 CO2 molecules, 10 NADH molecules, and 2 FADH2 molecules are produced in total. The next step involves electron transport in the electron transport chain, where oxygen is reduced by NADH and FADH2 to generate a proton motive force for the formation of ATP molecules. During cellular respiration as a whole, glucose is oxidized to produce 6 molecules of carbon dioxide, 6 molecules of water, and 36 to 38 molecules of ATP. Now that we understand the processes involved to produce energy, let's look at processes that require energy, such as reproduction. Both plant and animal cells go through similar reproductive processes. Gregor Mendel, the father of modern genetics, used pea plants to understand basic genetic principles about plants, and we've since found that these principles apply to animal cells as well. Also remember that in animals, we have somatic cells that undergo mitosis, and sex cells, or gametes, that undergo meiosis. So when we're talking about mitosis, we are talking about somatic cells only. In short, mitosis is where a diploid cell, or 2 copies of each chromosome, divides to form 2 identical diploid daughter cells. Meiosis is a process by which diploid cells undergo meiosis in 2 stages, meiosis I and meiosis II, resulting in 4 haploid gametes. Remember, diploid means that the cell has 2 copies of each chromosome, and haploid refers to each cell having only 1 copy of each chromosome. Animal cells have gametes that exist as male, meaning they have an X and a Y chromosome, and gametes that exist as female, meaning they have 2 X chromosomes. We refer to these as sperm for males and eggs for females. Both of these cell types will undergo meiosis I and II to form 4 haploid cells that will become diploid cells, or zygotes, once fertilized. For plants, meiosis also occurs in gametes, but it's not as straightforward. Plant cells alternate generations and therefore have a different cell cycle from animal cells. For plants, there are 2 generations, sporophytes and gametophytes. This differs based on the type of plant, but in general, most trees and grasses you would see when you look outside your window are in the sporophyte generation. So let's start there. Sporophytes are diploid cells that will undergo meiosis to produce haploid spores. These spores will then undergo mitosis to form the haploid gametophyte generation. This may seem weird to have haploid cells going through mitosis, since we just learned that animal cells have diploid cells that undergo mitosis to form more diploid cells. But remember that the point of mitosis is to produce genetically identical daughter cells, and in both cases, that's exactly what mitosis does. So we have our haploid gametophytes. The gametophyte will produce gametes for the plant cell, which are called sperm and egg, that are also haploid-like animal cell gametes. Pollen is a good example of the male gametophyte. So when the sperm gametophyte fertilizes the egg gametophyte, it results in a diploid zygote. From here, the zygote can undergo mitosis as usual to form more sporophytes. Both plant and animal cells generally undergo mitosis and meiosis in similar ways. The main differences between the reproductive processes of plant and animal cells are due to structural differences. Plant cells don't have centrioles like animal cells do. While centrioles act as an anchoring site to organize microtubules that aid in pulling chromosomes apart during cell division, they are not necessary for cell division to occur. The other main structural difference has to do with the fact that plant cells have a cell wall. During cytokinesis, instead of forming a cleavage furrow like animal cells, plants form a cell plate in the middle of a cell. This plate consists of components from the plasma membrane and cell wall that are delivered in vesicles. However, the big picture difference between plant and animal cell reproduction has to do with the fact that plant cells have a different life cycle than animal cells. Alright, now that we've fully compared and contrasted plant and animal cells, let's go over a review question together. of the following organelles, which ones are primarily found in animal cells? A. Ribosomes, B. Peroxisomes, C. Plasma membrane, or D. Centrioles? The answer is D. Centrioles. Both plant and animal cells have ribosomes and peroxisomes. Even though plant cells have a cell wall, they also have a plasma membrane like animal cells. With few exceptions, only animal cells have centrioles for mitosis and meiosis. The cell cycle is continuous and occurs so that cells can grow, regenerate, or repair themselves. Mitosis is one of the specific stages of the cell cycle where cells prepare to divide. In order to do this successfully, cells must evenly split their genetic material, or DNA, between each cell. Mitosis only occurs in somatic cells, meaning all the cells in your body except for sex cells. It's important to remember that before cells undergo mitosis, they replicate their genetic material so that each daughter cell has the same genetic material as the parent cell. Let's take a second to talk about chromosomes. Chromosomes are condensed strands of DNA. They're divided into two sister chromatids held together by their centromere. However, as we'll see next, DNA is only organized like this for certain parts of the mitotic process. So, to make this simpler, we divide mitosis into 4 phases that happen in the following order: prophase, metaphase, anaphase, and telophase, or PMAT if that helps you remember. Let's look at this a little closer. We start with a somatic cell, since that's the kind of cell that will undergo mitosis. Our cell is a human diploid cell, which means it has 2 sets of chromosomes, one set of 23 from our mother and one set of 23 from our father. This is true for all somatic cells in our body. Sometimes you'll see the shorthand for diploid as 2N. In order to maintain the number of chromosomes in our cells, the cell replicates its DNA before mitosis. So let's remember this as we look at the rest of the phases. During prophase, the cell's nuclear membrane dissolves and the chromosomes start to condense. Microtubules extending from the centrioles on both sides of the cell also begin to form. Together, they form a spindle apparatus called the centrosome that will eventually help move the chromosomes into place. Next, the cell will move into metaphase, where the microtubules attach to the centromeres of chromosomes, causing the chromosomes to line up along the equator of the cell. Now the cell is ready to enter anaphase. During this phase, chromosomes get pulled apart into their sister chromatids and move to opposite ends of the cell. The plasma membrane also starts to indent to prepare for the actual division of the cell. As a side note, we can now refer to these individual sister chromatids as independent chromosomes. Don't worry about this too much, but just be aware of the terminology here. Lastly, there's telophase. The chromosomes relax, the spindle apparatus disassembles, and the nuclear membrane reforms in each end of the cell. Lastly, the plasma membrane of the cell develops an even deeper furrow that will end up dividing the cell down the middle. The process where the cell actually divides itself in 2 is called cytokinesis. But this process stands by itself. Mitosis formally finishes up with telophase. Let's look at the end result a little closer. Remember how we started with a diploid cell with 46 total chromosomes? We've ended up with 2 diploid cells, also with 46 chromosomes total. These cells are called daughter cells and are identical to the one we started with. Before we go, let's look at a review question. During what phase of mitosis do the chromosomes line up along the equator of the cell? A. Prophase, B. Anaphase, C. Metaphase, or D. Telophase? The answer is C. Metaphase. All the microtubules attach to the centromeres of the chromosomes and pull them to the center of the cell so that we get an equal number of chromosomes when the cell undergoes cytokinesis.
Well, macromolecules are the biological molecules that make up cellular structure and make function possible. In this video, we will take a look at the structure, the function, and the source of the 4 macromolecules: carbohydrates, proteins, lipids or fats, and nucleic acids. Carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen. There are 3 classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the most basic form of a carbohydrate. Since they are the most basic form of carbohydrates, they cannot be further broken down into simpler sugars. They are distinguished by the amount of carbon atoms they contain, which is between 3 and rarely more than 6 carbon atoms. Monosaccharides that are usable for living organisms include glucose, the most common, galactose, and fructose. Disaccharides are made up of 2 monosaccharides. Examples of disaccharides include sucrose, maltose, and lactose. Polysaccharides are composed of a long chain of monosaccharides, as the name suggests. Examples of polysaccharides include starch and glycogen. How a carbohydrate is structured determines how energy is stored, and just how the breaking of carbohydrate molecular bonds let off energy. Carbohydrates supply our bodies with readily usable cellular energy. Glucose, the simplest carbohydrate, enters into our cells' mitochondria, is converted into energy in the form of ATP, and is used for various cellular functions. Carbohydrates are also used to tag lipids and proteins, which helps in immune function, cellular communication, and allow for cells to attach to the necessary extracellular material. Our central nervous system and muscles need a lot of energy in order to function, and this energy comes from carbohydrates. Consumable carbohydrates can be found naturally, occurring in fruits, vegetables, milk products, and grains. Proteins. Proteins are the most structurally complex biological molecules. Proteins are made up of chains of polypeptides, which are made up of amino acids that are connected together by peptide bonds. There are 3 types of proteins: fibrous proteins, which are structural, globular proteins, which are functional proteins, and membrane proteins, which are interactive. Fibrous proteins are mainly lengthy and narrow chains and are used for structural purposes. They generally have a repetitive sequence of amino acids and are more durable or less affected by temperature change, pH change, and so on. Examples of fibrous proteins include fibrin, actin, myosin, elastin, keratin, and collagen. Globular proteins are typically more dense and round-shaped, and their role is primarily functional. Globular proteins generally have a more irregular sequencing of amino acids, and they are going to be the less durable in terms of temperature and pH changes. Examples of globular proteins include insulin, immunoglobulin, enzymes, and hemoglobin. Membrane proteins, like their name suggests, are the proteins that are part of biomembranes or proteins that are interactive with biomembranes. There are 2 types of membrane proteins: integral membrane proteins and peripheral membrane proteins. Integral membrane proteins are the proteins that are permanently integrated within the membrane. They are often referred to as intrinsic membrane proteins because they are located within the core of the membrane. They are able to permanently attach to the membrane due to their hydrophobic side chains. Most integral proteins spread across the entire membrane. Peripheral membrane proteins are proteins that are only temporarily a part of a membrane, or integral membrane proteins. The way that they attach to the membrane is often indirect. They typically attach by interacting with an integral membrane protein. However, they can attach directly to the membrane, but it will be by interacting with a lipid polar head. Peripheral membrane proteins are also referred to as extrinsic proteins because they connect with the exoplasmic face of the membrane. So the particular sequencing of amino acids in a protein cause it to fold into 3-dimensional shapes. Well, the specific shape determines the protein's specific function. A protein has mechanical actions that gets paired with certain chemical occurrences, and this combination of motion and chemistry is what makes the function of proteins so crucial and dynamic. Protein function is dependent on attaching or binding with other molecules. There are specific portions of proteins and other molecules marked for binding, and these are called binding sites. This allows for proteins to function as catalysts, motors, pumps, signal receptors, or switches. Proteins are the building blocks for our body's tissue and can also be used as fuel for the body. Protein is found in effectively every part of our body. Protein is found in large amounts in meat and dairy products. It is also found in beans, nuts, seeds, and vegetables. Lipids, or fats. Lipids are characterized by their insolubility in water. There are 5 main classifications of lipids: fatty acyl glycerolipids, glycerophospholipids, sterolipids, and sphingolipids. Fatty acyl refers to fatty acids and their byproducts. It is made up of a hydrocarbon chain on one end and a carboxylic acid group on the other end. The hydrocarbon chain is nonpolar and hydrophobic, which means water-fearing. The end containing the carboxylic acid group is polar and hydrophilic, water-loving. Glycerolipids are also known as triglycerides. Triglycerides are composed of 3 fatty acids and a glycerol through the process of esterification, the reaction of carboxylic acids and alcohols. Glycerophospholipids are also known as phospholipids. They are composed of 2 fatty acids, a glycerol, and a phosphate group. Glycerophospholipids are a major ingredient of cell membranes. Sterolipids are a type of steroid with 4 attached carbon rings and a hydroxyl group. Cholesterol and cortisol are examples of sterolipids. Cholesterol helps to form the cell membrane where it serves as a secondary messenger in response to extracellular signaling molecules. Cholesterol also helps maintain proper cell fluidity. Cortisol is your body's primary stress hormone. It is most notorious for aiding your body's fight or flight response. Sphingolipids are a type of lipid with a foundation made up of sphingosine bases. Sphingosine bases contain 2 or 3 hydroxyl groups and generally a trans double bond in the 4th position. The most common types of sphingolipids in our bodies are dihydrosphingosine and sphingosine. Sphingolipids are known to protect the surface of a cell from harmful environmental factors. They are also involved in signaling and cellular recognition. Overall, lipids have crucial functions within cellular processes. This includes storing energy, communication, protection, and they offer structural stability. Saturated fats are common in animal fats and are solid at room temperature. Unsaturated fats are common in plants and are liquid at room temperature. Nucleic acids. Nucleic acids are composed of nucleotides. Nucleotides have 3 subunits: a nitrogenous base, a 5-carbon sugar, and a phosphate group. There are 2 types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. DNA and RNA get their names from the type of sugar they are composed of. DNA contains the sugar deoxyribose and RNA contains the sugar ribose. There are 2 types of nitrogenous bases: purine bases and pyrimidine bases. Purine bases have 2 rings in their structure. Adenine and guanine are the purine bases found in both DNA and RNA. Pyrimidine bases have only 1 ring in their structure. Thymine and cytosine are the pyrimidine bases in DNA, and uracil and cytosine are the pyrimidine bases in RNA. Nucleic acids make up a cell's genetic information. DNA works as an organism's instruction, and RNA works to make a cell's proteins. Nucleic acids can be found in vegetables, meat, as well as in yeast.
Hi, and welcome to this video on carbohydrates. Today we'll be looking at what carbohydrates are, what their purpose is, and what they're made of. So what is a carbohydrate? It's actually a type of macromolecule. Macromolecules are essentially groups of smaller molecules bonded together to carry out a specific function. There are 4 types of macromolecules, all of which have specific structural and functional properties that allow them to work together to carry out different cellular processes within the body. Let's take a closer look at these structures and see how we can use them for classification. Carbohydrates consist of 3 elements: carbon, hydrogen, and oxygen. They exist in a specific ratio of 1 carbon atom and 2 hydrogen atoms for every atom of oxygen. A simpler way to represent this is with the formula CH2O, N, where the N stands for any given number of these carbohydrate units. These simple sugars can link together to form more complex molecules called polymers. So carbohydrate monomers are the subunits of carbohydrate polymers. We can have simple sugars of just 1 monomer or more complex sugars with lots of monomers all bonded together. This means they vary in structure and are therefore really diverse. One way we can classify carbohydrates is by the number of monomers they contain. If the carbohydrate is made up of a single sugar monomer, it is called a monosaccharide, mono meaning one, and saccharide meaning sugar. Each of these are made up of chains of carbon, hydrogen, and oxygen in the CH2O N ratio, but they differ by N. Since N is directly related to the number of carbon atoms, we can also say they are classified by the number of carbon atoms present. A carbohydrate with 3 carbons is a triose, a carbohydrate with 4 carbons is a tetrose, and a carbohydrate with 5 or 6 carbons is known as a pentose and a hexose, respectively. These monomers exist as a ring structure, which makes them more stable. Pentose sugars like deoxyribose and ribose make up our DNA and RNA, so we definitely don't want those sugars to be unstable. Glucose, the primary sugar our bodies use for energy, is an example of a 6-carbon sugar with the formula C6H12O6. Carbohydrates can also be made up of 2 monomers called disaccharides. Some examples are lactose, which makes milk sweet, sucrose, which is your regular table sugar, and maltose, which is a product of starch breakdown. Each of these disaccharides is made up of 2 other monosaccharides. For example, sucrose is a glucose molecule and a fructose molecule bonded together, while maltose is just 2 bonded glucose molecules. When carbohydrate monomers bond together, they form covalent bonds through a condensation reaction. These links are called glycosidic bonds and are named more specifically by the numeric carbon to which it bonds. Carbohydrates that are made up of more than 2 monosaccharides are called oligosaccharides if they contain a few sugars, typically less than 10, and polysaccharides if they contain more than 10 sugars. So, polysaccharides are just complex oligosaccharides. Polysaccharides can be categorized based on the type of monomers they have and their structure. If a polysaccharide is made up of repeated units of the same monomer, it is called a homopolysaccharide. Contrastingly, a polysaccharide that is made up of repeating units of different monomers is called a heteropolysaccharide. Homopolysaccharides and heteropolysaccharides can exist in a simple linear unbranched form, or a branched form depending on the monomeric chemistry. Today we'll talk about the 3 main homopolysaccharides: glycogen, starch, and cellulose. Glycogen is made up of lots of glucose monomers that glycosidically link together to form a branched structure. A branch occurs every 10 glucose units of the chain. If your body has more glucose than its energy needs indicate, your body will store the glucose for later use in the form of glycogen. If your body needs to dip into its glucose store, it can easily do so by breaking glycogen down into glucose in processes like glycolysis. Plants, on the other hand, store their glucose in the form of starch. Starch consists of a mixture of 2 polysaccharides, amylose or amylopectin. Amylose is a long chain of glucose monomers, while amylopectin is made of branches of glucose monomers. Compared to glycogen in humans, starch is very similar. However, starch polymers are more spaced out than glycogen, meaning there's branching less often. Cellulose, the 3rd form of polysaccharide, is made up of lots of long chains of glucose molecules. The glucose molecules in the individual chains are linked by glycosidic bonds like in other polysaccharides, but the chains are also bonded to each other through hydrogen bonds. Cellulose is used mostly for structural support and can be found in cell walls. Sometimes a polysaccharide called chitin is considered its own class of polysaccharide, but really it has the exact same structure as cellulose. except it has an acetylated amino group at the second carbon instead of a hydroxyl group. Carbohydrates aren't limited to bonding just with one another. In fact, a lot of carbohydrates don't exist solely on their own, but instead form molecules called glycoproteins. Glycoproteins are found everywhere in the body and can be categorized based on their function. This dynamic duo is what gives our cells the ability to communicate with each other, gives our body a functioning immune system, and provides cushioning and support throughout. Glycoproteins are divided up into the three following groups: glycoproteins, proteoglycans, and mucins. The glycoprotein class has a variety of functions, but a good example is erythropoietin. Erythropoietin stimulates the production of red blood cells and enhances the stability of protein in the blood. The carbohydrate portion of this molecule is what makes up the ABO antigens to give us blood types. In general, glycoproteins are proteins that bind to carbohydrates embedded in or on the outside of cell membranes. They also act as markers to help other cells identify that cell, and they can aid in cell adhesion and structural support. The proteoglycan class is a protein attached to a glycosaminoglycan. The glycosaminoglycan's composition varies because it is made up of different repeating disaccharide units that contain amino sugars. Generally, proteoglycans are for structural support and lubrication. For example, a greckin in the cartilage of your heel will release water molecules upon impact to provide cushion for the surrounding muscle and bone. Whereas the glycoprotein and proteoglycan class are mostly made up of protein, the mucin class is mostly made up of carbohydrates. Mucins are responsible for hydrating cells and creating a protective barrier, so we mostly see this class of glycoprotein in mucus secretions of the respiratory, gastrointestinal, and urogenital tracts. Okay, now that we've covered everything, let's look at a couple of review questions. Which disaccharide is made up of one glucose molecule and one fructose molecule? Is it A, glucose, B, sucrose, C, maltose, or D, glycogen? The correct answer is B. Glucose is a monosaccharide. Sucrose is made up of glucose and fructose. Maltose is made up of two glucose molecules, and glycogen is a polysaccharide. What kind of bond is responsible for linking sugar molecules together? A, ionic bond. B, glycosidic bond. C, covalent bond. D, hydrogen bond. Or E, more than one? The correct answer is E. Sugar molecules are linked together via glycosidic bonds, which are a type of covalent bond. So both B and C are correct. There is some hydrogen bonding that goes on between the polymer, glucose chains, and cellulose, so D is also a correct answer.
Hi, and welcome to this Mometrics video about lipids. Today we're going to define what lipids are, learn about their physical properties, and then take a look at the structure of two important types of lipids. The International Union of Pure and Applied Chemistry, the scientists that decide on official names and definitions, loosely defines lipids as substances of biological origin that are soluble in nonpolar solvents. That's a pretty vague definition, and it has to be, because lipids are a diverse bunch of molecules that include fats, phospholipids, steroids, and certain vitamins. But we can pull a couple of important things out of this definition. First, lipids are of biological origin. In other words, they are naturally occurring molecules that have significant biological function. And second, lipids are soluble in nonpolar solvents, which is another way of saying that lipids do not dissolve in water. Since most biology is done in aqueous solutions, that's pretty rare for biological molecules and is the primary physical property that defines a lipid. Notably, this definition doesn't include any specific structural characteristics for lipids. So, even though steroids, water-insoluble vitamins, fats, and phospholipids are all considered lipids, they look very different from each other. However, they are each primarily made up of carbon and hydrogen, hydrocarbons. Let's take a minute to understand why hydrocarbons, and thus lipids, are not water-soluble. Hydrocarbons are generally water-insoluble because they are nonpolar molecules. In other words, there aren't areas of partial positive or negative charge because carbon and hydrogen share the electrons equally in their covalent bonds. This means hydrocarbons don't form strong interactions with water and therefore don't dissolve in water. For this reason, you'll often hear hydrocarbons called hydrophobic, which means water-fearing. Now that we have the general definition of lipids, let's focus on two specific types, fats and phospholipids. Fat is probably the lipid you're most familiar with. In fact, fat and lipid are often used interchangeably in everyday language. But as we just learned, in biochemistry, fat is actually a subgroup of lipids. Fats are also one of the 3 macronutrients, along with carbohydrates and proteins. They serve primarily as an energy source in the body, but also provide insulation and protection, as well as transportation for water-insoluble vitamins. Most fats are glycerides. Glycerides are a combination of glycerol and fatty acids. Glycerol is a small organic molecule with 3 alcohols, OH, and fatty acids are a long hydrocarbon chain, the fatty part, attached to the carboxylic acid, the acid part. The carboxylic acid allows the fatty acid to easily link with alcohols to form esters. Because there are 3 alcohol groups on glycerol, up to 3 fatty acids can attach to one glycerol molecule, which are called triglycerides. Most dietary fats we encounter, like butter, olive oil, and animal fat, are triglycerides. The hydrocarbon chains vary greatly in length, level of saturation, and geometry. In this context, saturation means that the carbon chain has the maximum number of hydrogens. In other words, the carbon is saturated with hydrogen. In an unsaturated fat, some of the hydrogens have been replaced with double bonds. The double bonds can have 2 different geometries: cis, where the carbon chains are on the same side of the double bond, or trans, where the carbon chains are on the opposite side of the double bond. The structural characteristics of the hydrocarbon tail determine the physical properties of different fats, like why butter is solid at room temperature, but olive oil is liquid. Let's take a look at one other type of lipid, the phospholipid. It's a diglyceride, meaning it's a glycerol linked to 2 fatty acids. In the 3rd alcohol position on glycerol, instead of an additional fatty acid, there is a phosphate derivative. This group is highly polarized and interacts very strongly with water. Thus, the phospholipid has a hydrophilic head and a hydrophobic tail, a property called amphiphilic. To maximize interactions between the phosphate group and water and minimize the interactions between the fatty tails and water, phospholipids form bilayers, a double layer of phospholipids with the phosphate heads pointed outwards and the fatty tails pointed inwards. This structure makes up cell membranes, the wall between the cells and the rest of the world, a pretty important job. The amphiphilic nature of phospholipids make them perfectly suited for creating barriers. Just another important reminder that function is always connected to the molecular structure. Let's quickly review what we've learned. Lipids are biological molecules that are water-insoluble, including fats, phospholipids, steroids, and some vitamins. While there is no specific structure that defines a lipid, they are all primarily hydrocarbon structures. Finally, we wrapped up by looking closely at the structures of fats and phospholipids.
To start, let's take a look at what DNA and RNA are, and then we will go back through and compare them. DNA stands for deoxyribonucleic acid and is a nucleic acid that contains the genetic instruction used in the development and functioning of all known living organisms. DNA is housed in the nucleus of the cell and works as a vehicle for storage and transferal of organisms' genetic information. DNA is a polymer that has a deoxyribose and a phosphate foundation with 4 nitrogenous bases: adenine and guanine, which are purine, and cytosine and thymine, which are pyrimidine. Now, adenine only pairs with thymine, and guanine only pairs with cytosine. DNA is a double helix structure and looks sort of like a spiraling staircase. The way that it spirals actually helps to give DNA added protection and stability. Let's take a look at exactly why. So, DNA has 2 strands, both located on the lateral ends, and these strands are twisted together like a spiraling staircase, which makes up the double helix. The lateral ends of these strands consist of sugar phosphate backbone of neighboring nucleotides that are bonded with one another. The phosphate molecule forms a covalent bond, which means it shares electrons with the deoxyribose sugar. These hydrogen bonds are what actually cause the DNA strand to spiral. The nitrogenous bases, adenine, guanine, cytosine, and thymine, are sandwiched between the 2 sugar phosphate strands. Each nitrogenous base is directly adjacent to another nitrogenous base, forming hydrogen bonds. Hydrogen bonds are weak bonds, but the bonds between the phosphate and sugar molecule are very, very strong bonds, which means they're very stable and can handle resistance. So basically, this phosphate sugar backbone's main job is to hold the nitrogenous bases that function as the coded set of instructions in place, and to protect them from outside elements. The spiraling of the sugar phosphate backbone adds 360-degree protection to the nitrogenous bases, rather than the nitrogenous bases being unspiraled and exposed. If it were not spiraled, then the weaker hydrogen bonds between the nitrogenous bases would be exposed to the surrounding elements, which could potentially break the bonds. RNA stands for ribonucleic acid and is a nucleic acid that executes the instructions given by the DNA. RNA is housed in the nucleus and the cytoplasm. The sugar in RNA is ribose. RNA is a single-strand chain of alternating phosphate and ribose units with the bases adenine, guanine, cytosine, and uracil bonded to the ribose. RNA molecules are involved in protein synthesis and sometimes in the transmission of genetic information. The primary role of RNA is to deliver the genetic information, or code, necessary for the building of proteins from the nucleus to the ribosome. This procedure stops the DNA from exiting the nucleus for the purpose of protecting the DNA. The production of protein is not possible without RNA. RNA is also crucial to the process of DNA transcription. An enzyme called RNA polymerase latches onto the DNA strand, then starts constructing a sequence of nucleotides to assemble a compatible RNA strand. There are 3 types of RNA: messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. mRNA is transcribed from DNA, and it transfers the coded in structure for protein synthesis. rRNA is a part of the ribosome and controls the translation of messenger RNA into the proteins. tRNA carries the amino acids that follow the correct coding to the ribosomes to be attached into the proteins. Now, let's review and compare DNA and RNA. DNA has the sugar deoxyribose, while RNA has the sugar ribose. The difference between ribose and deoxyribose are fairly subtle. Ribose has 1 more OH group than deoxyribose has, and that is the only difference. Though the contrast is subtle, the difference changes the entire function. DNA is double-stranded. RNA, on the other hand, is single-stranded. DNA is more stable under extreme conditions, but RNA is not very stable due to it only being single-stranded. DNA and RNA perform different functions in humans. DNA is responsible for storing and transferring genetic information, while RNA directly codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. DNA and RNA base pairing is slightly different since DNA uses the bases adenine, thymine, cytosine, and guanine. RNA uses adenine, uracil, cytosine, and guanine. Uracil differs from thymine in that it lacks a methyl group on its ring. Atoms are the building blocks of the universe. They make up everything you see around you. But what makes up an atom? At the very center of an atom is the nucleus, which is made up of small particles called protons and neutrons. Protons are very small, positively charged particles, and neutrons are neutral particles that have no charge. Atoms can have just one proton, or they can have multiple. A group of atoms that all have the exact same number of protons are called an element. For example, hydrogen is an element with one proton in the nucleus, and carbon is an element with 6 protons. In general, an atom will have a specific number of neutrons in the nucleus, meaning the atom won't lose or gain any neutrons for a very long time. This is called a stable atom. Usually, a stable atom has an equal number of neutrons and protons, but there are exceptions. To find the mass number of an element, you add the number of protons and neutrons together. So, protons plus neutrons equal mass number. This gives us names like carbon-12 or carbon-14, which are types of carbon atoms used in carbon dating. Orbiting the atom's positively charged center are particles with a negative charge called electrons. The electrons are attracted to the positive nucleus, but they can escape their orbit by an outside force. Atoms have a certain number of electrons orbiting the nucleus. If the number of electrons is equal to the number of protons, the overall electric charge of the atom is neutral. If that atom has more electrons than protons, its charge will be negative. With fewer electrons than protons, the atom will have a positive charge. The electrons determine how atoms interact with each other. Atoms can share electrons to form molecules, which are particles made up of many atoms. Water is made up of 2 hydrogen atoms and 1 oxygen atom. These 3 atoms share electrons. Now that we have talked about the different parts of the atom, let's summarize a few properties of atoms. Atoms have an atomic number, which is the number of protons in the nucleus. The periodic table arranges atoms in increasing atomic number. The charge of an atom is calculated based on the difference between the number of protons in the nucleus and the number of electrons orbiting the nucleus. Here's a quick review. An atom is made up of protons, electrons, and neutrons. The number of electrons will determine how atoms interact with one another and decide if the atom as a whole is positive, negative, or neutral. The mass number of an atom is found by adding together the number of protons and neutrons. And finally, the charge of an atom is determined by the number of protons and electrons in the atom. Have you ever put an ice cube in a heated skillet and watch it transform from ice to water, then to evaporate out of the pan and make steam? What you're seeing is that piece of matter changing in its state. Hey everyone, welcome to this video on the states of matter. There are 3 primary states that matter exists in: gas, liquid, and solid. However, in very extreme cases, matter can exist in other states, like plasma, more specifically, hot plasma. Gas. Within a gas, particles have large gaps of space, and there's no specific pattern to their arrangements. The particles move very quickly and freely. Since there's a lot of space between particles, gas is compressible. Gas can easily flow since particles dart past each other, and gas takes the shape and volume of the container that it's in, since the particles move freely. Liquid. Particles within a liquid are much closer together, but they also do not have a specific arranged pattern. These liquid particles move, but not quite like gas particles. They vibrate, slowly move about, and they can also move past each other. Because the particles can move, a liquid can fill the shape of the container it's in. However, it will not fill the whole space like gas, depending on the volume of water to volume of container ratio. Since there is not as much space between the particles, liquid is not easily compressible. Liquid can flow pretty effortlessly, which is due to the particles' ability to slide and move past each other. Solids. Now, particles within a solid are very, very close together with very little, if any, space between each other, and they are generally arranged in a specific pattern. Typically, solids do not move about, but they will vibrate in place. A solid has an already fixed volume and shape, since there is little to no major movements of the particles. A solid is also not easy to compress because of the lack of space between the particles already. Solids do not flow easily because of the lack of movement of the particles and their inability to move past each other. Now, plasma. I mentioned that this only happens in extreme cases. Plasma is extremely hot gas that is ionized. Plasma has significantly different characteristics than the other three states of matter, so it is thought of as the very distinct fourth state of matter. However, it is closest in similarity to that of a gas. Having an understanding of what's going on with the particles within matter is very important. They're not just simply tiny pieces of solid or liquid or gas, they are molecules and atoms. What is happening with those molecules and atoms, their physical characteristics, is what decides their state. Hey guys, welcome to this Metrics video over chemical reactions. In this video, we'll take a look at the definition of a chemical reaction, characteristics of chemical reactions, types of chemical reactions, and other things that are pretty important to know. First, the definition. A chemical reaction has taken place when a chemical substance alters its initial chemical identity and takes on a new chemical identity. So basically, what's happening is you have at least two molecules that come together perfectly, hitting each other in the exact right way, and these two molecules, change into something else. That's typically how all types of reactions work. For example, you have an eager and excited teacher who says, "Science is very important. You need to learn. So do your homework, do your homework, do your homework." Then let's add to the equation that one guy in the class who says, "I didn't do my homework." When these two come together and react, the product will be a very unhappy teacher and a slightly terrified young boy. In this example, we can see some observable differences after the reaction has occurred. Well, the same thing is true with chemical reactions. Every chemical reaction yields a different result, which inevitably means that chemical reactions will not all have identical observable properties. However, if you mix two chemicals together and want to know if they reacted with one another, you could look for these observable characteristics. Irregular change in color. If you mix white and red together and get pink or light red, then that's probably not a sign of a chemical reaction. That's just what happens when you mix the colors red and white together. However, if you mix a red and white substance together and get blue, then you might have a chemical reaction. Change in consistency. When two substances are mixed together and there's a change in consistency, then this may reflect a change in the molecular structure. So if you take two runny liquids and mix them together and the product is a thick jelly substance, then you might have a chemical reaction. Change in luster. Luster refers to the reflective quality of a substance. So if you mix two dull, murky substances together and they produce a really glossy or polished substance, then you might have a chemical reaction. Appearance of bubbles or froth. The formation of froth or bubbles may suggest that a gas has been composed in the chemical reaction. Change in odor. Now, if it's just a slight change in odor, it's probably not a chemical reaction. So if I mix a substance that smells like honey, now this is just a silly example, with a substance that smells like vanilla and the ending result smells like some type of syrup, then it's more than likely not a chemical reaction. If you take those two same substances and mix them together and their product smells like a smelly sock, then you may have a chemical reaction. Change in temperature. You can use the thermometer to take the temperature of each substance before, and if there's a change after the two have been combined, then there may be a chemical reaction. Precipitate formation. This just means that an insoluble solid is formed from a liquid solution. If this happens, then you probably are witnessing a chemical reaction. So those are some observable characteristics that may signify that a chemical reaction has taken place. Now let's take a look at 5 different types of chemical reactions. Number 1, combination reaction or synthesis reaction. A combination reaction is pretty straightforward. It can be defined as when substances are mixed together and they form a new compound. Here's what the combination reaction equation looks like. A plus B yields AB. As you can see, these two substances literally combine to form a new substance. An example of this is carbon solid and oxygen gas reacting together to form carbon dioxide gas. Number 2, single replacement reaction. A single replacement reaction, also known as displacement reaction, can be defined as a chemical reaction in which one element displaces or replaces, whichever word you would like to use, a comparable element in the compound, causing a chemical change in the compound. Here's what that looks like in equation form. A and B plus C yield AC plus B. As you can see in this equation, C has stepped in and replaced or displaced B. An example of this is the combination of copper and silver nitrate. 3, double replacement reaction. A double replacement reaction can be defined as a chemical reaction in which the ions of two compounds swap with one another within a liquid solution and configure two new compounds. Here's what that looks like in equation form. AB plus CD yield AC plus BD. An example of this is the combination of sodium sulfide and hydrogen chloride to produce sodium chloride and hydrogen sulfide. Sodium and hydrogen are cations, and sulfur and chlorine are anions. The cations and anions switch places during the reaction to form new compounds. Number 4, decomposition reaction. Decomposition and composition are opposites. In a decomposition reaction, a single complex compound is broken down or decomposed into a more basic substance. Here's what that looks like as an equation. AB yield A plus B. Now, there are actually 3 different subtypes of decomposition reactions: thermal decomposition, photodecomposition, and electrolytic decomposition. The term that precedes the word decomposition refers to the type of energy used to make the decomposition possible. So, in thermal decomposition, heat is added to the complex compound to decompose it. In photodecomposition, photons, or light, is added to the complex compound to decompose it. And in electrolytic decomposition, electric current passes through a liquid solution to decompose it. For example, the electrolysis of water. Water decomposes into hydrogen and oxygen when an electric current is added. Number 5, combustion reaction. A combustion reaction can be defined as a chemical reaction where a substance mixes with oxygen and produces substantial amounts of energy that take place in the form of heat and light. An example of this would be the combining of methane and oxygen to produce carbon dioxide and water. Now, the last thing we'll talk about, as promised, is other things that are pretty useful to know. There's a lot that I could go into here, but I just want to talk about 2 things. The first is basic language, and the second is reaction rates. Basic language. You should already know this, but here it is. The individual substances, before they are combined together to react, are called reactants or reagents. These reactants, once combined, yield a product or products. Secondly, reaction rates. Now, there's a lot that goes into this, but I'll give you the most basic idea of what this means. Chemical reactions are all over the place when it comes to speed or rate it takes for the reaction to occur. Some reactions happen instantaneously, and others take years and years. Well, reaction rates are dependent upon certain characteristics of the reactants. There are two variables in which the reaction rate is dependent on: the change in concentration of a substance and the time it takes for the change to be observed. Well, this rate of change can be altered via a catalyst, which speeds up the chemical reactions, or an inhibitor, which slows down chemical reactions. Understanding how chemical reactions work is important, but understanding the rate at which they take place is equally as important.
Hi, welcome to this video on balancing chemical equations. In any reaction, the law of conservation of mass must be followed. In other words, when reactants recombine or decompose to form products, we can't lose or create atoms. We have to end with the same number we started with. Chemists represent this in a balanced chemical equation, in which the number of each type of atom on the reactant side equals the number on the product side. Conversely, an unbalanced reaction has unequal numbers of each type of atom on the reactant and product side of the equation. For example, here's the unbalanced reaction of the combustion of methane. Methane reacts with oxygen to produce carbon dioxide and water. Now, while this equation accurately represents the chemical identity of the reactants and the products, it's unbalanced. If we count the atoms on each side of the reaction, we'll discover they are not equal. It's often helpful to keep track of our atom count in a table. While counting, remember that the subscripts, the 4 in methane, 2 in oxygen, 2 in carbon dioxide, and 2 in water, they denote the number of atoms of the element preceding the subscript within the compound. On the reactant side, we have 1 carbon, 4 hydrogen, and 2 oxygen atoms. On the product side, we have 1 carbon, 2 hydrogen, and 3 oxygen atoms. We need to balance this reaction. Note, to balance the equations, we have to honor the chemistry, meaning you can't simply add in extra compounds or atoms to achieve balance. This is because, of course, the equation must still reflect the chemistry that actually takes place. So, for example, while this equation is technically balanced in that there's the same number of each element on either side of the reaction, it's chemically inaccurate. This reaction doesn't produce H2 gas and certainly doesn't use a lone oxygen atom as a reactant. We also can't adjust the subscripts of compounds to achieve balance, as this would change the chemical identity of the species. So again, while this is a balanced equation, we are no longer representing the combustion of methane. O3 is ozone, and chemically it's very different from O2, and H4O is hydrogen hydrate, or H2 and H2O. By changing the subscripts, we change the chemical identity of the species, and we wind up with an entirely different and probably unviable reaction. Instead, to balance a reaction, we adjust the number of each species in the chemical reaction. This is represented in the chemical equation by adding coefficients in front of the chemical species. For example, if we put a 2 in front of water, this means we have 2 water molecules, or 4 hydrogen and 2 oxygen atoms. Now notice by doubling the water, hydrogen, in addition to carbon, is now balanced. Now to finish, we just need to balance oxygen. We have 2 on the reactant side and 4 on the product side. So by simply adding a second molecule of oxygen to the reactant side, we have ourselves a balanced reaction. Note that it's standard to drop the coefficient when it's 1. Now, we can read this equation as 1 molecule of methane reacts with 2 molecules of oxygen to produce 1 molecule of carbon dioxide and 2 molecules of water. Notice that the equation is also balanced with these coefficients. We've simply multiplied each coefficient by 2, which does give us a balanced chemical equation. However, we typically want the lowest whole number coefficient. So similarly, while this equation is also technically balanced, it uses fractions rather than whole numbers as coefficients. So once you're finished balancing your reaction, make sure there's no common denominator between coefficients or any fractional coefficients. If there are, either divide out by the common denominator or multiply to get the lowest whole number coefficients. Now that we understand how to balance equations, let's take a step back and think about why they are important. These coefficients not only ensure we follow the law of conservation of mass, but also dictate the stoichiometry of the reaction. You'll sometimes see the coefficients referred to as the stoichiometric coefficients. For example, in the combustion of methane, to produce 1 mole of carbon dioxide, we need 2 moles of oxygen, but only 1 mole of methane. Put it another way, the mole ratio is 2-to-1 for oxygen and carbon dioxide, but 1-to-1 for methane and carbon dioxide. Thus, these coefficients are necessary for calculating limiting reagents, yields, molarity, and other calculations that require converting between moles of species within a reaction. Basically, it's pretty important to have your chemical equation balanced correctly. Though there is no one surefire way to balance chemical equations, there are a few useful techniques to apply. So, to summarize, it's usually best to balance the elements in the more complex compounds or multiple compounds. Do that first, and then leave simple species for the latter steps. Second, for elements in only one species on either side, try using the subscript of the opposite side as a coefficient. Third, while neither of our examples dealt with charged ions, if you do encounter a chemical equation with ions, make sure that the charge is balanced as well. After all, you don't want to lose electrons. Lastly, some equations are simply going to be more challenging than others. It's important not to get frustrated. Just jump in, start by balancing one element and evaluate how that changed the entire chemical equation. Then move on to a second element, add a coefficient, reevaluate. Keep running through this process until you've got yourself balanced equation. Remember to check that you've found the set of lowest whole number coefficients. Now that we've pointed out a few tips for balancing reactions, try some more practice. Pause the video and balance the following chemical equations. Oxidation of nitric oxide by oxygen, roasting of the sulfide ore, pyrite FeS2, combustion of ethane, and decomposition of lead nitrate. All right, hopefully that went well. Here are the 4 chemical equations balanced. The last 3 were pretty challenging, so don't feel discouraged if they took you a while or if you weren't able to solve them. The best thing to do is keep practicing. You'll start to feel more comfortable with the process. That's it for today's video. Thanks so much for watching and happy studying.